So this is Ralph Moss, and today we have the great honor of talking to Dr. Thomas N. Seyfried, who is professor of biology at Boston College. Professor Seyfried received his Ph.D. in genetics and biochemistry from the University of Illinois in Urbana, I guess we pronounce it, in 1976. He's best known for his work with metabolism and ketosis, and how it intersects with diet and illnesses such as epilepsy and, of course, cancer. Professor Seyfried is the author of about 200 peer-reviewed articles listed in PubMed, and about half of those specifically concern cancer. So, Professor Seyfried, thank you so much for joining us today.
Well, thanks very much, Ralph. It's nice to be here.
So, you remember that I came and I visited you in Boston, Chestnut Hill, I guess it is, at your lab in, well, it must have been about 8 years ago. And
Wow.
The occasion of that was your publication of your book, Cancer as a Metabolic Disease. And I've had a chance to read that book many times. I've referenced it so many times in my own writing, and looking at it again in preparation for this discussion, I really must say that I think that this is one of the most revolutionary works that has ever been published in the cancer field. Offhand, I can't think of many other books, and I am a student of the history of cancer and of cancer research, and not too many that really go to the root, to use the word, the original origin of the word radical, and go to the root of the problem and really try to globally change thinking about this disease. And I think the book has stood up very well over the past 10 years. In your book, you make some, what I would say would be surprising, even astonishing statements about cancer. And so I want to ask you about these and give you a chance to explain them, because they're going to sound odd to or contradictory to a lot of our listeners. They are some cases the diametrical opposite of the so-called conventional wisdom on cancer. But I think people will really enjoy hearing from the expert on what this means and why you've come to these views after almost 40 years in the field. And if possible, of course, try to put it in language that lay people can understand, because that's our audience is for the person who's dealing with cancer, who's seriously interested in understanding the basis of this disease and its possible. So the first thing that you say that strikes me as being new to most people is the idea that some, if not most, conventional anti-cancer agents largely operate through reduced caloric intake. This is a strange notion to most people. Could you explain what you mean by that?
Well, I can't be sure whether all conventional treatments work through that process, but the concept came to me. I'm working with Dr. Purna Mukherjee, who got her PhD in calorie restriction, and she and I have been working together on this problem for 20 years at least. And when we were doing some original work with a drug called N-butyl deoxynitromycin, it doesn't make any difference what the name of it is, but it was supposed to stop ganglioside biosynthesis, and which are complex glycolipids. You hear a lot about that from Tay-Sachs disease. But cancer cells have abnormalities in these gangliosides, and I got my PhD in ganglioside biochemistry. So we had a drug that blocked ganglioside biosynthesis. A company, pharmaceutical company, had given us this drug to look at changes in gangliosides. And when we put it on, we do things. We put it on some mice that had some tumors. We were looking at other things besides looking at Tay-Sachs disease and things like this. But we had some tumors growing in the lab, and we gave this drug to the animals, and it shrunk the tumors tremendously. And of course, the drug company got very, very excited about this, as well as inhibiting ganglioside biosynthesis. Um, so obviously, it looked as though we have a drug that would stop ganglioside biosynthesis and at the same time shrink the tumor. Well, this became, a massively interesting drug to this company, and they had ideas of patenting this drug and doing all this kind of stuff. And then Perna and a couple of other people said, "Well, you got to be careful about some of these drugs because if the drug is working through a unique mechanism like targeting ganglioside biosynthesis, you have to be sure that the drug isn't also influencing some external event, something outside what you think it might be. Like maybe so." We were watching the mice. It was very interesting. They were eating. We had to mix the drug in with a powdered chow, and they were eating the powdered chow at the same level as the control group that did not get the drug, but they were losing body weight. And I said, "Wow, this is interesting. The animals eating the drug were losing body weight." And I said to Pern, I says, "Do you think the therapeutic benefit of this drug is not through inhibition of ganglioside biosynthesis, but rather through an inhibition of body weight? In other words, the drug was acting like something that would reduce calorie." And we put another group of mice in after we saw this phenomenon. We put another group, a control group of mice in that did not get the drug, but we took food away from them so that their body weight matched exactly that of the group that was getting
Uh-huh.
the drug. And what we found is the tumors shrunk to the exact same size as the animals that were getting the drug and were losing body weight. And then when we looked at the ganglioside composition, of course, the ganglioside composition was significantly reduced in the drug group, but wasn't reduced at all in the calorically restricted group. So consequently, the drug had yes, it was inhibiting ganglioside biosynthesis, but it had not. But the inhibition of ganglioside biosynthesis had nothing to do with the therapeutic benefit of why the drug shrunk the tumor. And
Oh,
then we later found out that the drug blocked sucrase activity in the intestines. And what was happening, the mice were eating this food, but they weren't able to digest the food. So it was like an indirect calorie restriction. And then we went on and looked at many of the other drugs that are being used in pharmaceutical, like temozolomide. Well, we gave mice temozolomide, and yeah, it killed the tumor, but they shrunk. That it also caused massive body weight. As a matter of fact, we couldn't starve mice enough to lose body weight as fast as temozolomide worked. So temozolomide, which is used in the glioblastoma brain cancer
yes.
field quite a bit, um, it seemed to increase basal metabolic rate. So even if our control mice, if I took all the food away from them, they could not drop body weight as fast as the group that was getting the temozolomide.
That's amazing.
so, so what I'm thinking, in fact, indirect calorie restriction is one of the mechanisms which we think is responsible for some, not all, of course, for some of the therapeutic benefits of some of these drugs. So,
I
so
see.
why give drugs now? We don't need to do this anymore because we now know that all cancers rely on glucose and glutamine for growth, and all we need to do is restrict glucose and glutamine. We restrict, and this will kill the majority of cancer cells because they're one disease. It's all cancers are the same, and they can't live without fermentation mechanisms. So when you calorie restrict, you're shutting down glucose availability, which is shutting down their glycolytic fermentation process.
Yeah, we'll
And
get into that.
but
Um,
I'm just saying, the
yeah,
bottom line is that you're absolutely right. I can't be sure how many successful cancer therapeutic drugs are working in part through indirect calorie restriction, but I wouldn't be surprised if many of them do.
I recently spoke to a patient who had a glioblastoma, brain cancer, and was taking Temodar or temozolomide is the scientific name, and he was losing weight, and it was strange because I didn't know this. What you just said. I was trying to figure out, was this person in cachexia? Was he, in kind of a free fall in terms of
Yeah.
his weight? But perhaps now I know something about that drug, Tavador, that it could actually, the drug itself could be causing that weight loss. And you say that alone is a kind of anti-cancer agent, but of course you can't starve yourself to death. Are you going to? I mean, even if a person stopped eating, and there have been over the years, and in fact, I'd say over the decades, attempts made to kill cancer by basically by water fasting. But at a certain point, you kill the person by doing that, right?
Well,
And so that's not
well,
a strategy in itself.
you have to do it the right way.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the General Scientific Concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
There's Guy Tannenbaum. He's all over the web. He had advanced prostate cancer.
Hmm.
He has a lot of things on the web now,
Hmm.
and he had like you. He had come to my office, and he was in a 20-day. He came with his son, so he had already been on a 20-day water only fast.
Hmm.
Um, and I talk about this stuff, but I don't often meet people who are actually doing it, or
Yeah.
actually sitting
Yeah.
in my office who haven't eaten for 20 days. Um,
Yes.
but he was diagnosed. He's a French. He speaks French. He has English and French productions on the web, and he wanted to let me know that he just did a series of 20-day water only fasts. Of course, in between time, he was eating small amounts of very highly nutritious foods, but then he would go on these 20-day fasts, and went from 130. Correction, 230 pounds down to, when I saw him, he was like 160. He looked great. Um,
Hmm.
he had advanced metastatic cancer into the bones, and where
Hmm.
prostate cancer goes. And
Hmm.
now he's completely without cancer. And the strange thing that he always asked me, he said when he went back to the oncologist never really asked him, "What are you doing?" It was like, well, of course. I mean, here's a guy. He should be right. He should be dead. And you think the oncologist said, well, "What are you?" And then he says, I'm doing water only fasting." And the oncologist said, "Well, I would continue to do that if I were you." So you can see now. I have to be honest with you. This is pretty draconian way to get rid of cancer. But this guy is, eating now healthy vegetables and all. He's on the web. And my other friend, JJ Tracon from France, he has a book coming out also. JJ Tracon, battling advanced metastatic kidney cancer, and his battle was horrific. He's the only pilot in air. He's a pilot. Air France flies the big jets, and he's one of the only person to get his license back after suffering advanced metastatic cancer. And
Hmm.
he managed his cancer using um, ketogenic diets with water only therapeutic fasting, and primarily a non-pharmacological approach. But it's very hard to bite the bullet and do these kinds of things. Our research is designed to adapt some of this with specific drugs in our press pulse strategy, so we can manage these cancers without as much food restriction as some of these other folks have done. But the
Hmm.
bottom line is the tumor cells need the glucose to survive, and with these water only therapeutic fasts are really pulling the glucose down, and starving out these tumor cells. They can't compete with normal cells when you put the whole body under a stressful condition. The issue is
So
the
what?
glutamine, and we're working on that
Yes,
now.
I was going to say that. So cancers, we know that about 90% of cancers will light up on a PET scan because, presumably because the FDG that's injected into them is a kind of quote unquote non-digestible form of glucose, of sugar, right? And so
Right,
you can
right.
see where the cancer is because after about an hour, um, the FDG contrast agent starts to accumulate in the areas that crave or they're avid for
Yeah.
glucose. But so I guess in the naive view, the view that I probably had before I read your book, um, you would think that if you just deprive the body of glucose, that you would then kill off 90% of cancers. But it seems like it's more complicated than that because there are other sources of nutrition. If the cancer is deprived of the glucose, or even if it's not, I mean, it could still be using the most abundant free amino acid in the body, which is glutamine, and perhaps other things as well. So this is sort of the crux of the matter. If you get rid of the theoretically, if you get rid of the glucose, or at least bring it down to a level that doesn't feed the cancer, what do you do about the glutamine?
Well, the glutamine, we can target that. There are drugs that are working on glutamine. We use the drug DON, dor deoxynorlucine. It's a glutamine analog,
Hmm.
and what it does, it shuts down the ability of the cells to use glutamine. It's a pan-glutaminase inhibitor.
Hmm.
People have used that drug in the past alone. Um,
Hmm.
and what happens is you get some responses that are really good and other responses that are really bad, but they never shut the glucose down when they use the DON.
Hmm.
So we published a big paper just last year showing how if you use a calorie restricted ketogenic diet to shut down the glycolysis pathway and then include the DON, you slaughter these cancer cells. That you have to realize glutamine is metabolized to glutamate to alpha ketoglutarate. There is no energy involved in the use of glutamine. Glutamine is generating energy in the TCA cycle, the Krebs cycle, at the succinyl CoA ligase step. It's a fermentation mechanism because all cancer cells have defective oxidative phosphorylation. We've shown this. Others have shown this. You have to look under the electron microscope to see this, but when you do in tissue preparations, um, so when and all other amino acids, it's like you said, maybe another amino acid, but every other amino acid needs to use energy to metabolize its core structure to alpha ketoglutarate, which is the key precursor for mitochondrial substrate level phosphorylation. So here's the situation: glutamine is the most abundant amino acid in the body. It's a
Hmm.
pure gold. It's pure gold to a metastatic cancer cell. So one of the characteristics of metastasis and cachexia, you get a lot of cachexia. Muscles start wasting away. The muscles are a great source of glutamine. So the cancer elicits the glutamine out of the muscles, and the patients, they eat food that looks like they're wasting away because the
Yes.
cancer is sucking down the glutamine. So in order to manage cancer, you have to do two things. You've got to shut down the glycolysis, the glucose availability, and you have to take away the glutamine. So when you do a calorie restricted ketogenic diet, you lower blood sugar, and don't forget the ketones are toxic to the tumor cells. So they're not only an alternative source of energy for the brain and heart and other organs, they're actually toxic to the tumor cells. But the normal cells thrive on ketones. So you're then shifting the body over to a completely new metabolic state, and then you come in with these glutamine inhibitors and you blast the hell out of these tumor cells without toxicity to the rest of the body. It's not that
Well,
complicated. All you have to know is the
but
biology of the problem.
but the drug Don that you talk about, I mean, I know you use that in your experiments, but that's not available to consumers or even to doctors, as far as I know. Am I wrong about that?
Yeah, no, I bet the pharmaceutical companies are building other things that are like that. As a matter of fact, the group at Johns Hopkins, they have a drug. They've modified Don a little bit. It's the same drug, but they might be able to patent that. And it's under
What
clinical trials at Johns
is that
Hopkins.
three bromopyruvate?
No. 3-bromopyruvate is a glycolysis inhibitor.
Mhm.
So you don't have to worry about. I mean, you can use 3-bro. It's very toxic though. The only problem with
Yes,
3BP is it's got a lot of
yes.
toxicity.
Yes.
Um,
So
why?
so you what
I mean,
we have a
so the
drug, metformin. I mean, metformin would block the uptake of glucose to a certain degree, wouldn't it?
it's not that effective.
Uh-huh.
I tried all this stuff.
Uh-huh.
It's okay. I'm not going to say people shouldn't take it. I mean, it's not like it's not your earth, it's not your earth, it's
No.
not your prime mover. We know what works. We know how to work the system. It's a strategy, and we published it in the press pulse concept,
Could
where
you
we
explain could you explain the press pulse concept, please?
yeah, the press pulse. Well, that was Aaron's a paleobiologist actually come up. He said the eradication of organisms on the planet always happened when you have two unlikely events happening at the same time. And basically, and this was the responsible for these mass extinctions that we had in Earth's prehistory. So you have some sort of a chronic stress on a population. It's strong enough to weed out the weak, but the stronger organisms kind of hang on. And then you have some sort of catastrophic pulse, like a meteor strike or a volcano explosion, and it wipes out everything. So
Hmm.
you already have a weakened system by a chronic stress, and then you wipe out the whole system with an acute pulse. So what we do, we just simply adopted that concept to the management of cancer. So what we do is we take humans and mice and whatever, and we bring them into a new metabolic state and we put chronic stress on the tumor. And as you said, Ralph, you can't kill all the tumor cells by just taking away glucose because some of them will hang on using glutamine.
Hmm.
So the concept of bringing the body into a new state is we remove systemic inflammation, which is important for driving the tumor with the press uh, calorie restricted ketogenic diet. We call it ketogenic metabolic therapy. The patient starts getting really healthy, but the tumor cells are hanging around. They're not like all going. They're not dropping dead all over. And then we come in with the glutamine inhibitor. So when the patient is in therapeutic ketosis, low glucose, elevated ketones, you then come in with the drugs and low doses. You don't have to be these high doses because once the patient is in therapeutic ketosis, we showed that these drugs work even better at lower dosages. You just need a little bit more to chronically, acutely kill off the survivors. And you do this without harm to the patient. The toxicity is minimal, if at all, and you just gradually degrade these tumors, switching from chronic stress to pulse stress and back and forth. And the next thing you know, either the tumor shrinks to very small levels, or it disappears completely. And we've seen this in so many people. They say, "Where's the clinical trials on this?" Well, you can't do a double-blind crossover on pulse press. You want to live, or you want to be part of a clinical trial. So it's your choice. I mean, I don't. I mean, it's up to people.
Yes. But again, I'll come back to the question of how do you attack the glutamine source? Because again, the DON or the DON, I couldn't find a source for that, and I don't know. I've seen some I've seen some writings on the internet
Yeah,
saying that maybe these anti-worm medications, mebendazole and
yeah,
fenbendazole, might
right.
act as inhibitors of glutamine. Also, I've seen some indication, and I had some confirmation of this, that Dr. Stanislaw Brzezinski's antineoplastons might
Yeah,
act as blockers of glutamine uptake. But what, practically speaking, and I'm sure that 90% of our listeners are in one way or another dealing with cancer or have it in somebody in the family or want to prevent recurrence. Practically speaking, how do you do this? I mean, is there a diet that people could use other than this, the fasting? But is there
yeah,
something that practical that people can do to be able to attack the sources? In other words, is, to quote from Jane McClellan's book, is there really a way to starve the cancer cell?
yeah. Well, you're right about the glutamine issue and Don. There are a number of companies. Cayman will produce Don. It's hard to get. It has to be delivered IV, intravenous as well. So
Hmm.
you it's a little bit more. Not that physicians do intravenous treatment all the time. I mean, this is not a medically difficult situation.
Yeah.
The problem is Don is not approved. I don't even want to say it's not improved because it had been used prior to on people with cancer. There are some, many studies published where Don was used, but it was never used together with targeting the glucose. But you're 100% right. Where do you get this? There are a number of companies in China that make Don. The problem is I've invalidated, whether all the products from these companies are good or not. I mean, you have to do it. I do it on a preclinical system before I recommend it for anybody. I mean, we get the drug from Sigma Scientific Corporation. I mean, if I had cancer, I would get it from Sigma or someplace.
But it's against the law. It's against the law to
listen, you want to keep your ass alive? I mean, who cares?
Yeah.
I mean, right? I mean, you have to be practical about it. I mean, we have evidence. I mean, we already showed that when you administer the drug with the ketogenic diet, we delivered 2 to 3-fold more drug through the blood-brain barrier when it was administered with the ketogenic diet. I mean, give me a break. I mean, they're trying to figure out all the ways to get through blood-brain barrier. You just put it on a ketogenic, and we did it with a different kind of a drug too. So ketogenic diets facilitate transmission of small molecules into the brain to target brain cancer. It's just that simple. Yeah, we published this. Now, what's the mechanism? We don't know. But the bottom line is we've quantitated using mass spectrometry to show that there is more drug going through the blood-brain barrier. So you don't need the high doses that you used to need to get in there, right? So that's why I'm saying Press Pulse works as a beautiful synergistic interaction between the different kinds of therapies. You're right. Now, there are a number of drugs that the pharmaceutical industries are producing, trying to mimic the effects of DON. Okay, there's a CB 833, or they got these numbers for these things. I'm not sure if they're as powerful as DON. And as I said, the Johns Hopkins group is doing a lot of work on a DON type analog. I think these will come. The problem is they just use that. So they go out and do a clinical trial using just the glutamine inhibitor, and they say, well, it's marginally effective. Well, you're not targeting the glucose, man. You got to target both. What's the matter? And then they say, well, we can't do that because we won't know if it's our drug that works or whether it's this that works. And we're interested in keeping people alive without toxicity. We're not interested.
The same old reductionism of we have to study one thing at a time, and it may be a synergy between different things.
Yeah, well, see, we've developed here at Boston College the single best series of animal models for cancer, systemic metastatic cancer, glioblastoma, all natural. These things are not these genetically engineered things that are artificial, not this xenograph, taking human cells and grow them on the ass of a mouse. I mean, this kind of stuff is it's not natural stuff. I mean, not to say that these are bad models. They just don't provide you want a model system that's going to replicate what goes on in the human. Okay. And this is what we have. So we know that the model systems that we have, when we see it working in these mice that we develop, we know it's going to work in humans, and we have seen it do this. The problem now is what we do to keep mice alive as long as we can are very difficult to do these experiments in clinical trials. Only because we have a press diet and we pulse with drugs at certain frequencies, and to do people know what it's hard to do a double blind on these kinds of things. So but the bottom line is if you have historical controls like GBM, where most of the people are dead within 2 years,
glioblastoma.
Yeah, glioblastoma. And if you have guys that are living 5 and 6 years on metabolic therapy, then you might want to figure out what the hell, why are they doing so well? We have Pablo Kelly over in England. He's now 6 years out with no radiation, no chemo, just metabolic therapy. We have other people, Andrew Scarborough. We're getting numbers of people. Alison Gannett. More and more people are coming on board because they want to stay alive. And I wrote a big paper clearly with Joe Maroon, team surgeon for the Pittsburgh Steelers and neurosurgeon at UPitt, showing that the standards of care for glioblastoma are largely responsible for the rapid recurrence and demise of the patients. You shouldn't ever irradiate the brain. You shouldn't irradiate people's brains. I already showed it creates fusion hybridization and sends these tumor cells out. The use of Avastin or bevacizumab should be banned. This is a drug that contributes to yet they use it all the time. It contributes to massive invasion through the brain. And temozolomide, it's like, what are you doing to these people?
Hmm.
We can manage these cancers so much better on metabolic therapy. But my colleagues in the field are yoked. They can't do it. They're not allowed to treat patients with metabolic therapy until all other therapies are shown not to work. And I'm saying, well, your damn therapy's not going to work. So why don't you come up from, oh, we can't do that. The IRB won't allow us to do that. And we're yoked. So these poor folks, the patients are trapped in a system that's a failed system, putting their lives at risk for the maintenance of procedures that don't work. It's like it's a tragedy, monumental tragedy.
Yes. And we hear a lot about ketogenic diet, but I think certainly from the time that you wrote Cancer as a Metabolic Disease, you were always saying it's not just ketogenic diet, it's also calorie restriction. But that's a lot harder for people. I mean, unless their back is against the wall, the idea of combining the cutting back on their food. We're such a consumer society that we like to take things as opposed to telling
Well,
people
well,
do
it,
less.
it, but it has to do with the species. In the mouse, when we fed the mice ketogenic diets ad libitum, that means unrestricted, and we had a ketogenic diet that they liked, they would scarf it down. They would get insulin insensitivity, and the tumors would grow much faster. So we would have to calorie restrict this. Human beings naturally calorie. The hormone cholecystokinin is an anti appetite hormone. It stimulates the 10th cranial nerve to shut down appetite. So when you eat a bunch of fat, oftentimes humans just don't. They stop eating. This is why the ketogenic diet is used as a weight loss diet. So
Yeah,
people are eating ketogenic diets, they're losing weight. And they say, how can I lose weight by eating all this fat? Because you're turning on cholecystokinin and shut down your appetite. So that happens more in humans than it does. Humans do a hell of a lot better on this than the mice do. So we have to be very careful in the mice eating too much ketogenic diet. Whereas in humans, they don't eat much. They have a lot of good food sources, very nutritional avocados and different kinds of
hmm,
foods that have high fat content, low carbohydrate content, and you just measure your glucose ketone index. And this is what we also developed here at Boston College was the glucose ketone index. So it allows people to know the ratio of blood sugar to blood ketones to know whether or not they're in therapeutic ketosis. And this can vary from one person to the next. So some guy might eat one thing and get into ketosis. Another guy might eat something very different, get into the same ratio. It's when you're in that ratio is when the chronic stress goes onto the tumor cells. And once you're in
I see.
therapeutic ketosis, yeah, then you go on, you take a drug that will target the glutamine and you work it that way. And I think this is going to be the future. This is what I just told you is the future. It's just, it may take people screaming and kicking to go into it. But, bottom line is people want to live. That's it. I mean,
That's for
there's
sure.
motivation for life. I mean, let's be
Yes.
honest.
So the ketone bodies, as they're called, these are just to explain for the lay listener. These are chemicals that are produced in the body when there's an absence of carbohydrates for the generation of energy. So the body has a fallback mechanism, right, to produce
Yes.
these ketones. Now you're saying that not only do these serve as an alternative source of fuel for normal cells, but that they're actually toxic to cancer cells. How does that happen?
Well, what we think is that they generate reactive oxygen species in the tumor cells, and the ROS kills them. Um,
Hmm.
all we know is that we and others have tried to grow cancer cells in ketones, and they die. And not only ketones, fatty acids as well. If you cannot, cancer cells need glucose and glutamine. You can't. Fatty acids and ketones cannot replace glucose and glutamine. And even what we find is even in the presence of glucose and glutamine, the ketone bodies can be toxic to the tumor cells.
I see.
Now, you have to have a good normal cells burn ketone. We evolved as a species. In fact, we as a human species, we evolved to starve. Our history on the planet has been one of constant starvation. So we have adopted we have selected in our genomes incredible means of storing energy. We store energy in the form of fat, but we also burn. Our history was not at McDonald's hamburgers and Taco Bell and all these other things that we're pounding in. And the obesity epidemic is only the result of our incredible history of being able to store energy. So
Uh-huh.
we're in a new environment now, which is producing type 2 diabetes, obesity, cancer, all these other kinds of diseases, based on our incredible ability to store energy because we evolved over millions of years as a starved species. Nobody was really fat.
Yeah.
You get your fat from eating bone marrow, or sweet maybe during particular periods of the season when you have a ripe fruit or a berry or something. But our ability to survive was based on burning fats, and fats themselves are broken down to ketone bodies because the brain can only use ketones or glucose. It cannot use fatty acids. So in order to keep our brain functional, when we're not taking in carbs, we burn fat. The fat goes to the liver, and the liver makes ketone bodies, which are small water-soluble fat breakdown products, and they can provide the brain with all the energy that we need. Um, but you have to have a good mitochondria in the cell to burn the ketones for energy. And we now know that all cancer cells have defective mitochondria. They can't burn the ketones.
Mm-hmm.
And they can't use fatty acids either. You need a good mitochondria to generate energy from fatty acids. So what cancer cells do is they store fatty acids. They form these little droplets. And in fact, if you look at under the electron microscope at tumor cells, and even sometimes under the light, you see all these vacuoles, these storage vacuoles. And you're saying, what the hell is going on? They're storing the fats because that's the way to protect them. They protect themselves because the fats will kill them if they're trying to go into they'll cause too
Oh,
many reactive oxygen species. So they
so
store the fat in these little vacuoles in the cells. It's
they're
called
isolating. They're sort of
Yeah,
isolating the fat. So,
yeah,
in this sense, maybe I misread Jane McClellan's thesis, but it seemed as if one of the ways that she maybe disagreed with you was emphasizing that cancers need fatty acids. If I read her correctly, and we did an interview
yeah,
with
yeah.
her,
No,
uh,
well,
y-
this is a
you're
kind
saying
of a
that's not true.
yeah, no. It appears that way that cancers need fatty acids. The issue is that the fatty acids stimulate glutaminolysis and glycolysis. Can you believe
Uh-huh.
this? So we
Yeah.
showed we have a paper coming out. So it looks like, oh yeah, cancer cells need fatty acids. Well, the fatty acids aren't used by the cancer cell directly. The fatty acids stimulate the tumor cells to take up more glucose and glutamine. So it's an indirect effect of the fatty acids.
I see.
So a lot of you'll see a lot of papers published in the literature saying, oh yeah, fatty acids are used by the tumor cells, and they're sucking down glucose and glutamine. Well, you take away the glucose and glutamine and give them the fatty acids and see what happens. They croak. So a lot of people don't do the right kinds of experiments. They think tumor cells have normal respiration when they don't. Because if you look under the electron microscope, they have no cristae, the mitochondria all screwed up. They're not going to be able to burn fatty acids or ketones. People have to come
Mhm.
to realize that. Otherwise, the ketogenic diet should make tumors grow 10 times faster. They don't.
Yes. So um, basically, all cancers are defective, or in terms of the mitochondria, which are the energy producing elements within the cell. And if I remember, our mutual friend Pete Peterson at Johns Hopkins, who wrote the introduction to your book, saying that on average, cancer cells have half as many mitochondria as normal cells. So it's
Yeah.
basically what you've sort of gone back to Otto Warburg, 1924, uh, basically locating the origin of cancer in the mitochondria. Am I correct?
Yeah. The thing about Otto Warburg and his colleagues is they were focused. His argument was that all cancer cells ferment glucose because their oxidative phosphorylation is defective. And people then went off and said, "Oh, but all these cancer cells are taking in oxygen and making ATP in the mitochondria. Therefore, Warburg must be wrong."
Yeah.
What we have found is that the energy coming out of the mitochondria, the few mitochondria that might be present in cancer, is not coming from oxidative phosphorylation. It's coming from mitochondrial substrate level phosphorylation, driven by glutamine and through the glutaminolysis pathway. Warburg did not know about this only because that mechanism was not discovered until the very end of his life, or
Hmm.
towards the end of his career. Um, and and even Warburg himself made the error of thinking that when tumor cells take in oxygen, that oxygen is producing ATP through oxidative phosphorylation in the electron transport.
Which to explain is the normal method by
Yeah,
which
yeah. You know,
normal
yeah,
cells produce energy.
right. All of it. We're breathing. Everybody's breathing.
Yeah.
And we breathe because we need the oxygen as the final acceptor for the electrons in the electron transport chain, so we can generate ATP.
Hmm.
Um, but the cancer cells, if you look at them in culture, you say, "Wow, look at these cancer cells! They're taking in oxygen just as much as a normal cell. Therefore, they must have normal oxphos." Wrong. We showed this. They're taking in oxygen, using it for different reasons. Other scientists have shown this as well. So where the hell are they getting their energy from? So they're getting their energy from glutamine. It's coming through and it's making massive energy in the TCA cycle. It's a substrate level phosphorylation. Here's another thing, Ralph, which has been recently found, that many tumor cells have the pyruvate kinase M2 isoform in the cytoplasm. They don't, in other words, it doesn't make ATP. You can make lactic acid, but you're making metabolites for growth, but you're not making ATP. Without
ATP
energy,
being
nothing.
the main source
Yeah, energy.
of packets of energy, right?
Energy is energy. You can't live without, nothing on the planet can live without energy. Cancer cells can't live with energy. We can't. You drink cyanide. Let's see how long you live when you drink cyanide. You shut down the whole system. You die.
Hmm.
All right. So where the hell are the cancer cells getting their energy from if it's not through glycolysis and it's not through the electron transport chain? Where the hell are these cells getting their energy to live? It comes from mitochondrial substrate level phosphorylation. That's why when you target glutamine and you've taken away glucose, these cells drop dead fast. And you can do that. And this is the new thing. The new thing that people don't understand is that the cancer cells are getting the majority of their energy from mitochondrial substrate level phosphorylation, giving the appearance that they might be using oxidative phosphorylation, but they're not.
Hmm.
Cancer cells can grow in cyanide. We can't grow in we can't live in cyanide. Why the hell is a cancer cell living in cyanide? Because it's
Interesting.
fermenting. It's fermenting.
Yes.
It's not using,
So it doesn't need oxygen.
no, doesn't need oxygen at all.
Right. Right.
So, and Warburg showed that, and he poured cyanide on his tumor, so couldn't believe it. So, I mean, you have a person with metastatic cancer, and they drop dead of a heart attack. I mean, their body is dead, but the tumor cells in that body are still alive. They'll be alive for maybe a couple of days.
Mm.
So,
Mm.
as long as they
Yeah.
can get fermentable fuel.
Amazing. So the takeaway message here is that it is possible to control cancer by controlling the food supply basically to the cancer. And the main things to worry about would be glucose, meaning uh, carbohydrates that would turn into increased blood glucose, and glutamine, which is the common free amino acid. Is there some, other than fasting, is there some diet that would minimize the amount of glutamine that's available?
No, but glutamine can be made from other sources. So
Hmm.
it's that's why they call it a non-essential amino acid. You can make
Hmm.
glutamine from glucose. However, when you're
Hmm.
shutting down glucose, you're also restricting, at least in the brain, you're also going to be cutting a little bit back on the glutamine. But glutamine is such an ubiquitous amino acid, like everywhere, and it's also
Hmm.
involved. It's extremely important for the urea cycle, as Hans Krebs showed. It's extremely important for the function of our immune system. The Newscombs from Oxford University in England showed that glutamine is the main fuel for cells of our immune system. We also know, for our gut
Hmm.
health,
Hmm.
a lot of cancer
Hmm.
patients are given high dose glutamine to help repair and the damage to their body.
Yes. Yes.
So it has a double edged sword on that molecule. You got to be careful because at the
Yeah.
same time we need the molecule, we also know it's driving our cancer. So, and people now need to know, and there's no diet that I know of that will restrict glutamine. So it's got to be that's why we use press pulse. The diets are okay for shutting down glucose, but then we have
Hmm.
to use certain low dose drugs to work together for this
Hmm.
ketogenic metabolic therapy using press pulse strategy.
What would those drugs be?
Well, those, like I said, Don, and listen, believe me, the pharmacy, here's the situation. You can't patent Don because it's a pan. What I mean by it's a pan glutaminase inhibitor. So it inhibits multiple glutaminases simultaneously. What the big companies would like to do is produce a drug that would do what Don can do. The problem is they haven't been successful yet in developing that kind of a drug. And many
Hmm.
of the drugs that they have tried are toxic. They're too toxic.
Hmm.
So, but believe me, this I would say that looking for the glutamine inhibitor that's non-toxic is probably one of the big hidden secrets of the cancer pharmaceutical. I mean, obviously they're using all these immunotherapies and CAR T. That's nuts too, in my mind, because it's based on the somatic mutation theory, and we know that the somatic mutation theory is now under serious reevaluation. So you have to have therapies that are based on the underlying biological problem of the disease. I mean, I'm not saying that immunotherapy, PD-L1 inhibitors, and CAR T immunotherapy, and these kinds of things don't work on some people, but they kill people too. They're very expensive. They can be incredibly toxic. So why are you doing that when you don't need to do it? This is the crazy thing that I can't find. I mean, what are you
Hmm.
doing? You understand the biology of the problem? Why are you doing that? Why are you doing all that nonsense? It's like, give me a break.
Well, I mean, the drugs are phenomenally profitable, 8.8 billion for Keytruda last year, um, but they're being given at a much too high a level. If you reduce the dose to about a third of what's currently being done, you don't lose any of the effectiveness. In fact, you increase the effectiveness and you reduce the serious side effects to about 1/10 the level of what's currently existing. So it's a money driven thing, but there is some truth to the fact that you
Yeah,
can
of course.
mobilize the immune system. But
Yeah.
I mean, for me, the takeaway message is what you just said. When most people, of course, they're focused on what can they do now vis-a-vis their own cancer, but it's also, we can't. As Albert St. George used to say, you can't really solve a problem that you don't understand.
For the first time,
Yeah.
putting them all together, where everybody could see the commonality in replacing bad mitochondria with good mitochondria and managing the cancer, regardless of the mutations in the nucleus. And then we went on. Now, new papers are coming out showing that there are some cancers that have no mutations. All right.
Hmm.
Now the new stuff is coming out showing that you have all these driver mutations in normal cells that never develop cancer.
Hmm.
Um, then we have the chimpanzee, our closest biological relative, 98, almost 99% identical in genome and protein sequence. And there's never been a documented case of breast cancer in a female chimpanzee. Yet they're genetically the same as we are. So, you put that together and you say, oh, wow, we have cancer cells that have no mutations. We have normal cells with driver mutations. We have chimpanzees that never get cancer. And you say to yourself, I mean, how in the hell are you able to say that cancer is a genetic disease? It makes absolutely no sense.
Are you passionate about the groundbreaking and heroic research of Dr. Thomas Seyfried on metabolic therapy and cancer? So are we. That's why we've created something special for you, in collaboration with Johnny Rockermeyer, a German book publisher and translator. Introducing our collection of meticulously crafted books that distill the essence of Dr. Seyfried's work. Dive into the science and discoveries. These summary books are your gateway to understanding the intricate world of metabolic ketogenic therapy in a clear, concise, and engaging way. Whether you're new to the subject or a seasoned enthusiast, our books offer insights that can change your life. Ready to explore this transformative knowledge? Visit our website at www.cancerasametabolicdisease.com to get your copy. You can buy the ebook there directly and the paper book via the provided links. Here's the best part. A portion of every purchase goes directly to support Dr. Thomas Seyfried's groundbreaking research. That's why the direct ebook purchase is the best option to donate as much as possible. You can see all of the donations Mr. Rockermeyer has already made at www.ketoforcancer.net. That's right, when you buy our books, you're not just investing in your own knowledge, you're also contributing to the future of cancer research. Help us make a difference. Together, we can drive change and save lives.
And anybody who wants to sit down and think about that you have to, if you have a rational thinking mind, you have to come to the conclusion there's no way in hell cancer can be a genetic disease. So we put it all together, and now we realize, and without energy, these tumor cells can't live. Where do they get their energy from? From fermentation. What drives fermentation? Glucose and glutamine. Well, why don't we focus on the glucose and glutamine issue? And when you do, you get results that are much better than anything that's currently being used in the
Yeah.
cancer industry.
I have
It's
one.
not
I have
that
one more.
complicated.
I have one more question for you, which has always been on my mind, and that is that Craig Thompson, who's the president of Memorial Sloan Kettering, is also in this field, sort of in terms of glutamine and metabolism, and he and his and Memorial Sloan Kettering patented a glutamine PET scan. But they never commercialized it. They have the patent on it, and I'm not making conspiracy theories here. I'm sure they must have run into different problems and so forth. But my question is this: if they would ever develop the glutamine PET scan, in other words, to see where there was increased glutamine uptake, the way they can see whether there's increased glucose uptake, what do you think it would show? I mean, do you think that tumors are predominantly glucose utilizing machines and not glutamine? Or do you think that a lot of tumors are doing both increased uptake of glucose and glutamine? Or are some tumors, as I suspect, some tumors are mainly just dependent on glutamine? For instance, cancers that are growing and don't light up on a PET scan, pretty much you can assume, right, that those are going to turn out to be glutamine dependent. But where's the line? Is it that in other words, what I'm asking is, theoretically, do you think it would be possible, if you could severely cut down on the amount of glucose going to the tumor through the various means that exist, to bypass the glutamine question entirely?
Yeah, I mean, these are very important questions, and the answer is that it depends on the cell. Some tumors will be more glutamine dependent, and others will be more glucose dependent. But in my mind, both fuels are used. We have some tumors here that are more glutamine dependent than glucose dependent, and we have just the reverse on some other tumors. And that, I think, is what the cancer field has already determined, that you have oftentimes there's very few tumors that we know of that are purely glucose or purely glutamine dependent.
Uh-huh.
Um, uh, Tumors that have mutations in the TCA cycle, which in other words block or disrupt the glutaminolysis pathway indirectly, forcing that cell now to use more glucose. They become more glucose dependent.
Uh-huh.
But majority of the here's the situation. We have two things. The glucose is primarily, the carbons of glucose are serving as the building blocks for the synthesis of proteins and amino acids and lipids and these kinds of things, so the cells can grow. The energy that drives the process is primarily coming from glutamine through the mitochondrial substrate level phosphorylation, as well as the glutamine nitrogen. The nitrogen is needed for DNA and RNA and protein synthesis. So the amide nitrogen on glutamine is used as a precursor for growth, along with the carbons of glucose. So the carbons of both of these molecules are distributed through the cell for the rapid growth, synthesis of the various building blocks that they need, DNA and RNA and protein synthesis. So it's a combination of needing both fuels for the proliferation of that cell. And I look at it as a purely energy, because every cell has a pump on the membrane surface. It's called the sodium potassium ATPase. It burns ATP in order to keep a gradient of ions across the membrane. And that allows the cell to be alive. And life means you're not at equilibrium. And to remain away from equilibrium, you need energy. And when the energy to the pumps fail, the cell swells and dies. Okay, it's not that complicated. So
Mm-hmm.
where the hell is the energy coming to maintain the pump activity? Whether you're a cancer cell, brain cell, any kind of a cell, you need to maintain the activity of the pump. So it's all energy. So if I pull the plug on the energy, the pumps will not get the ATP and they'll fail and they'll kill. So the tumor cell is dependent on glutamine for energy and glucose for growth. So you pull the plug simultaneously on glucose and glutamine, and the problem becomes manageable. But
Yes.
yet there's not a single clinical trial anywhere on the planet that's simultaneously targeting glucose and glutamine.
It's unbelievable.
It's
Unbelievable.
unbelievable.
It is. It is. Well, listen, I hope we can continue this discussion some point in the future, and tremendously appreciate you coming on the podcast, and I'm sure that the listeners will too. Talking to Professor Thomas Seyfried of Boston College, who's done the remarkable work of showing how cancers are dependent on glucose and glutamine for energy, and how that could be used as an effective cancer treatment. So thank you so much, Tom, for agreeing to be on the show.
Thank you very much, Ralph. Real pleasure.